Thank you for the invitation and also thanks for the kind introduction. It is my great pleasure to be here to talk about something I'm pretty interested in for a while. So that is how we're gonna combine randomized clinical trial and the real world data for treatment effect evaluation, how to improve the treatment effect evaluation process. And the two data sources actually act like they are in couple therapy. So they have different backgrounds, different habits, sometimes do not speak in the same language. But causal inference, we are like the therapist trying to convince them if they work together, they can actually build a healthier, more robust evidence base. And we can actually achieve a lot by doing data integration. And today I'm gonna talk about one of the topics. How we're gonna enable long-term treatment effect evaluation by combining short-term RCT, randomized clinical trials, and long-term follow-up in real-world settings, and using the so-called semi-parametric proximal DID framework. And here's the usual disclaimer. The project is supported by the US FDA U01 grant, and the contents are our own, not representing any official view from FDA or the government. And this work is developed in close collaboration with industry partners, especially colleagues from Eli Lilly, including Ming Yang, Wendy Elia, and also students from NC State, Young Park. He is actually doing all the heavy lifting. And our motivation actually is very practical, addressing challenges in post-approval evidence generation and a long-term effectiveness assessment. So to set the stage, I'm gonna step back a little bit to put our work in a broader landscape of evidence generation, especially how policy has acted like the catalyst to promote the use of real-world data and the real-world evidence. And then I will talk about the interface of RCT, RWD or E, and the causal inference, how we're gonna combine the conventional wisdom and the new technologies. And the topic of proximal DID framework fits the direction, the topic very well, in the sense that we're gonna estimating the long-term treatment effect by combining the two data sources. And then in the end, I have some summary and thought for the path forward. So the research actually is motivated by this 21st Century Cures Act passed a long time ago in 2016, which is designed to help accelerate medical product development and new innovations to patients faster. It emphasizes two things. One is big real-world data, and the other one is precision medicine. And in response, the US FDA has developed a framework called the RWE framework, which stands for real-world evidence, trying to evaluate the potential use of real-world data and real-world evidence for drug and device approval. And since then, there's many government guidance and initiatives to support the use of real-world data and real-world evidence. Especially a long time ago, we know like the drug approval require like phase 3 randomized clinical trials, right? And how we're gonna leverage now like the emerging real-world data and real-world evidence to speed up the process. So there are guidance and considerations to the industry how they can like submit documents, use real-world data and real-world evidence to FDA. And not just the US FDA, so you can see some guidance on the left that's produced from the US FDA government. But there are also considerations and guidance from European. So in the middle, you can see some EMA guidance, UK guidance, and also guidance from China and Japan. So the global picture is very clear, okay, very similar, right? So they are trying to embrace the emerging trend of using real-world data and real-world evidence. So this is quite active agenda for research as well. So let's first understand real-world data and real-world evidence a little bit more by contrasting the RWD and RCT, the conventional RCT. Okay, we know that in typical settings, randomized clinical trial is the gold standard for treatment effect evaluation, right? So that's required by the FDA if like some sponsors want to approve some drugs or devices because we have randomization of the treatment. So it can ensure internal validity. We can minimize biases, right? We can compare apples to apples. But there are also some limitations associated with RCT. So for example, in RCT, usually you have very restrictive enrollment criteria. And therefore, the underlying population may not represent the target population well in clinical practice, right? So for example, only 8% of cancer patients actually participate in oncology trials. Okay, so the trial population may be limited in patient diversity, right? Representativeness. And also, due to the practical reasons, RCT may be small in size, follow-up can be short, and the cost is high. But for real-world data, it is opposite, right? Because we have this real-world data collection mechanism. You can get access to like disease registries or the EHR systems. The underlying population is more representative of the target population in clinical practice. There's a rich diversity. We have the sample size large, follow-up long, and cost is relatively cheaper compared to running a randomized clinical trial. But we know that RWD are not collected for research purposes, and therefore there are many sources of biases, right? And then, for example, you cannot control all the confounding variables that may bias your result for the treatment effect, right? And then we need to apply standard state-of-art causal inference methods for causal inference of the treatment effect. But you cannot guarantee all the confounding variables has been captured in the real-world setting, right? So this is the limitation associated with using real-world data for causal analysis immediately. But from the table, it is very clear that the two data sources actually have complementary features, right? So the future is not about replacement, but integration. So we can actually achieve a lot by doing data integration. So there are some key objectives in data integration. I listed a few. So for example, we can improve the generalizability of RCT findings because we want to apply the RCT findings to a broader, more diverse patient population, right? And secondly, we can improve the efficiency or statistical power of RCT analysis by leveraging some information from real-world data, right? There are tons of information in real-world data out there. Why not we borrow like some strength, right? And then thirdly, we can enable long-term treatment effect evaluation, which is not plausible in a lot of cases from RCT alone, because RCT usually is designed to answer short-term questions, right? Long-term follow-up actually is very expensive, and many others. But we need to be cautious because real data may have different sources of biases. So FDA has already documented a few bias sources associated with real data. For example, selection bias. That means the RCT and the RWD population may be different, right, in terms of measured confounding variables. So in this case, we can observe the biases and we can use statistical method to deal with the bias. But there may be unmeasured ones, right? Unmeasured confounding, some key disease characteristics are missing in either data source, then we need to handle this unmeasured confounding. Lack of concurrency, that means different studies may happen in different times, right? And there may be some change of the effect that occurs over time. So we need to handle this in certain studies. In measurement errors, data quality issues, right, in real-world settings. Usually, these are real-world studies not designed for research purposes, right? And therefore, we need to handle tons of data quality issues and outcome validity issues. Sometimes in different data, how to define the variables, the definitions are different. So we need to be aware of this. So although RWD, RWE are very promising, we must develop a statistical method that can handle bias as well. Okay, so that's a key point here. In terms of data integration, I think the causal roadmap are very helpful because we need to be clear what the steps for the data integration. So in the very first beginning, we need to define what is the causal question of interest, the causal parameter of interest. So we call this causal estimate. And then we invoke certain causal assumptions so that we can link the statistical parameters with the causal question, right? So it translates to a statistical problem. Then we estimated the statistical parameter with the right tool, okay? So the causal roadmap helps us to clarify the inferential goals and estimate in the data integration context, whether the target population is the RCT population or the combined population or just the external population of interest, right? And then it also helps us identify and address biases in different data sources, whether it's confounding, selection, or measurement error, data quality issues, and we can articulate assumptions and identification strategies so that everything becomes very transparent. And currently, we know like AI, ML are very trending. Here is another like integration, how we're gonna combine typical statistical foundations with the AI, ML methods. And in causal inference, we actually think that AI and ML can be embedded in some like doubly robust estimators. So that means we can leverage the predictive power of AI ML for us to model like some nuisance parameters because they are good at modeling complex relationships or the high dimensional relationships, right? And at the same time, we want to deliver statistical rigor for the ATE estimator. And doubly robust estimators actually offers such an avenue. So this is why I emphasize the statistical foundations. These are the statistical principles that can offer such like advantage. Okay, we can still use AI ML for prediction, but we can also like deliver very transparent statistical properties for the ATE estimator. Okay. And along the years, I actually developed this causal inference framework for combining RCT and RWD. By the time we wrote the review paper, we divide the research into two lines. One is how we're gonna leverage the representativeness of real-world data to improve the generalizability of the RCT findings. So we kind of like go from RCT to RWD. We want to like generate the RCT findings to the RWD population, right? So we can do this for a lot of estimates of interest, for example, average treatment effect, ATE, ITR that represent individual treatment regimes, which is a key concept in precision medicine. And in certain cases, RWD only have summary statistics, and we can also do some certain degrees of generalization in this context. Still, like we can leverage the summary statistic only from the RWD to make a generalization possible. Okay, so this is the first line of interest. I actually came here a few years ago. I actually talked one of the topics along this line. But later years, I actually developed some other research lines of interest. For example, the second bottom. Here, we want to go the opposite direction. So for a lot of cases, RCT is of small size, right? So we want to improve the efficiency or the statistical power of the RCT analysis by leveraging rich information in real-world data, right? So we want to borrow strength, borrow information from real-world data. But the key is we don't want to borrow bias, right? We only want to borrow useful information. Otherwise, you're going to bias your result. So bias strategy, mitigation strategy actually is very important. So we have developed like test and pool strategy, bias modeling and correction strategy, and also selective borrowing. So these tools can help us identify or detect the bias and correct the bias from the borrowing procedure. So this is quite important and quite interesting. But the field is evolving very quickly, and there are many other more objectives we can do. And today I'm gonna talk about a recent one, how we're gonna estimate the long-term treatment effect from short-term RCT and long-term real-world data. Okay. So why we are interested in long-term treatment effect? This is because this long-term treatment effect evaluation is quite important because they determine whether the treatment has sustained efficacy, whether there's adverse events accumulate later on, and whether the therapy is cost-effective or not. And these are very important for health technology assessment. So if you are interested in pharma statistics, like this determines the health technology assessment you may encounter in real world, like a drug submission process, okay? In long-term RCT usually are not feasible. This is because RCT usually are designed to answer short-term questions, right? Longer follow-up will require like higher cost. And also there may be like logistic burdens and ethical issues like patient crossover. So in this plot on the right, this is a typical scenario. So for the RCT, we have a trial period. And at this primary endpoint, the treatment has been demonstrated to be effective. Okay, and then it is allowed for the RCT control or ethically required for them to switch to the treatment, right? Because the treatment already have been shown to have some short-term efficacy, right? And then you can see still for the treatment group, we can observe some long-term outcome in the extension period. But for the control, this long-term follow-up actually is missing, right? We don't observe such outcomes anymore for the control setting. But if we want to evaluate the long-term treatment effect, we need to estimate this, right? We need to fill this gap. So this is a critical evidence gap. And the central question is what will happen to those RCT patient population over the long term if they have not switched to the treatment, right? So this is the counterfactual we want to estimate. And we have some external data that provide the long-term control outcome, but the question is whether we can directly borrow such information, right, to fill this gap. So this is the problem of interest. And we can not naively augment RCT with external data because we face some challenges. I listed 3 here. The first one is unmeasured confounding, right? Because we may have like some key patient characteristic missing, and that characteristic can influence both the study participation and also health outcome of interest. For example, the baseline health consciousness, right? And underlying disease severity. And if we ignore such unmeasured confounding, we can bias our result, right? So that's the first challenges we are facing. The second one is study heterogeneity, okay? You can imagine that the experience of being in the trial is very different from the experience of in the real world settings, right? For example, in the trial, you may receive more care, you are monitored more closely, right? And then there are also placebo effect or the so-called Hawthorne effect. So that means the outcome trend will be very different even for the identical patients if the person is participating in the trial or just in the external cohort. Okay, so this is the study heterogeneity we need to handle. The third one is informative missingness because in the extension period, patient dropout is very frequent and the dropout is really random, right? You can imagine sicker patients may be more likely to discontinue. And if this is the case, the remaining sample actually is a biased sample, right? So the remaining sample actually is a healthier patient population compared to the original patient population of interest. So we need to handle the informative missingness in this case. And ignoring any one of these can bias our long-term treatment effect. Okay, so we need to handle 3 challenges here. And there are many existing approaches available. They have been developed over the decades. So for example, standard data integration use extrapolation or predictive powers, predictive models, synthetic controls, matrix completion, difference in differences, proximal inference. So I will mention these ideas later on. But the message here is, although they can handle certain parts of the challenge, for example, unmeasured confounding or study heterogeneity or informative missing, but there's no methods available to handle all 3 challenges simultaneously. Okay, so we need a unified framework to overcome confounding, heterogeneity, and the missingness. And we introduce this proximal causal inference with a flexible DID structure. So I will also later mention what they mean, like capital MAL, PDR. These are the key components in our framework. But here I want to emphasize addressing the 3 challenges are quite important because we want to align directly with the current regulatory guidance. And there are 3 guidance. One is FDA RWE framework on the use of like fit for purpose data. Okay, and then the ICH E9 estimate framework. So in this framework, we need to define the target population and also how we're going to handle missing data well. And then the FDA's like the guidance on how we're gonna handle the missing data, like the informative missingness. So addressing the three challenges are quite important in terms of the regulatory guidance. And here is the typical regulatory use case. So just want to put our context in real world settings. We start with the phase 3 randomized clinical trials. We have a very strong internal validity in the sense that the RCT trials are randomized. And we have limited follow-up due to the practical reasons. And then crossover happens after the primary endpoint. Okay, so we don't observe long-term control outcomes, okay, for the RCT patient population, but external control data are available, but we also may observe informative dropout during the extension period, okay? And then our objective is to estimate the long-term treatment effect in the original RCT patient population, okay? And here is our statistical framework. We first describe what is the data structure and then introduce the potential outcomes. So how many of you heard about potential outcomes in causal inference? Yeah, many of them, right? But later I will emphasize that these potential outcomes are a little bit different. We need to make sure that we capture all the interventions of interest. So we start with the data structure. So we have two data sources, and we introduce S to be the steady indicator. So S equals 1 if the subject is coming from RCT, and 0 if the subject coming from external control. X is the baseline covariate, and now we have two time periods, the pre-extension period and the extension period, which is represented by this pre and the 0 respectively. And then for each individual, we observe a treatment sequence, A pre, A0, an outcome sequence, Y pre, Y0, and observation sequence, R pre, R0. Okay, and then we abbreviate A0, Y0, R0 as AYR, respectively, just for simplicity. U is unmeasured confounding that can affect the study participation and outcome of interest. Okay, and we want to make this explicit that we have an unmeasured confounder U. Okay, now to define the causal parameter of interest, we introduce this potential outcomes notion. Okay, so YT as A pre A0, this is the potential outcome, has subject been assigned to study as and the treatment sequence A pre A0. Okay, so I want to emphasize that the intervention here, we actually have two kind of intervention. One is study participation and one is the treatment sequence. So even like for some patients, you have received the same treatment sequence 0, but participate in the RCT or external cohort, maybe another intervention, okay? So even like with the same treatment 0, so all control, we still have another intervention. Okay, I think that introducing this study participation as an intervention is key to address the study heterogeneity I just mentioned, right? Because in the experience of being in the trial may be fundamentally different from experience in the real world setting, okay? So just remember that we have two interventions of interest, okay? And for these potential outcomes, we also need to make this a stable unit treatment value assumption so that all the potential outcomes are well defined. Okay, for example, the future treatment does not affect the past potential outcome. Okay, and we also make the causal consistency assumption. So for the YT, the observed YT in external control, actually it's the potential outcome, okay, 0, 0, 0, right? So the study participation is 0, okay, in this case. And the YT 0, 0 here, that means this is the potential outcome here, okay, where S is the actualized study participation, okay. So implicitly, I'm gonna make all these super assumptions. In using the potential outcomes, a good thing is we can define the causal estimate of interest very clearly. Okay, these are mathematical construct, but I think in this notion we can define what is the parameter of interest, right? Here we are interested in long-term average treatment effect for the RCT patient population at the extension time. So you can see I'm contrasting the two outcomes, the YT, the trial treated and the trial control for the trial patient population, okay? And it will be very clear here. So I'm interested in long-term, right, potential outcome, the trial treated outcome and the trial control outcome. This one is observable, but this one is missing due to crossover. Okay, so this part, the one part of the estimate actually is very clear. So this part actually is observable. So it is identifiable directly. But this part, because of the crossover, this potential outcome is missing. So the challenge lies in identifying and estimating this parameter when we have a long-term follow-up for the external control. Okay, but you can see like these potential outcomes are different due to the study intervention, okay, 1 versus 0, right? So that's a challenging part, and I'm gonna ignore all the treated part because there's no controversy in identifying and estimating this part, and if we're gonna only focus on the controls, like from the RCT and the external control, okay, from the rest of the talk. And to make the discussion more concrete, I introduced this motivating example, which I will use throughout the talk. So we have an RCT that is called the SOLA trial that examined the effectiveness of SOLA on slowing the progression of ultimate diseases, together with some external control that's from the JERUS observational studies that provide the long-term follow-up for the external controls. And we want to combine them so that we can estimate the long-term treatment effect. And we have a rich set of covariates, 19 in total. We have BMI, age, duration of diagnosis, and also several cognitive and functional scores. And importantly, we treat this baseline MMSE score as a latent disease severity. Okay, so this is treated as the unmeasured confounder U. Okay, and the outcome of interest is the instrumental activity of daily living, so IADL score, measured across 4 time points, the baseline, the week 28, week 52, and week 80. Okay. In the trial, actually, we follow the patient through week 80, but we intentionally treat the crossover happens at week 28. Okay, and we are interested in the RCT control trend at week 52. In this design, we actually have some like benchmark, right? So we actually follow the RCT patient through week 80, so we intentionally treated crossover there, and therefore we can compare our methods with the benchmark. Okay. So this is the plasma simulation we conducted. So we use the actual data to fit this study inclusion probability and also potential outcomes, and then generate the data and then intentionally treat the crossover happens at week 28. Okay, so this is the proximal simulation. So you can see we have unmeasured confounder U that affect both the study participation and also the potential outcomes. Okay, and then also for the potential outcomes, we have this study heterogeneity. That is the interaction of the study participation and also the covariate X and U here. Okay, so that means if you look at the mean trend for the external control and the RCT control, they do not overlap, right? So there are study heterogeneity. Actually, this is the marginal trend. If we examine the conditional trend given covariates, they are also different because we have the unmeasured confounder U. And they enter the potential outcome for the external control and the RC control differently, right? So even for the conditional outcome mean given covariates, they are different. So that's the challenging part, how we're gonna recover the external control trend from RCT control trend from the external source, right? Okay, so here we intentionally treat the RCT control actually crossover at this time point, and we are interested in the target here. Okay, so that's the RCT control outcome mean at week 52. Okay, so in truth, we know this, right? And then how we're gonna leverage this observation and this observation to recover that. Okay, so that's our objective.
What is the outcome variable?
Outcome variable is the IADL score.
Cognitive and functional score?
Yes.
So those are part of the outcome?
Those are not part of the outcome. They are separate measurements, but they are correlated.
But the thing is,
they are highly correlated.
What
potential outcome definitions, right? So when you consider the A3 and A0 vector, why are we only interested in 1, 1 and 0, 0 combination? Why can't we have 0, 1, that kind of a pairing, right? So your AT definition had,
Yeah.
you see the 1, 1 and then 0, 0 difference, right? So
Yeah.
can't we be interested in somebody previously not treated, then getting treated, like a 0, 1?
Yeah. Yeah, you can focus on that question. So we are answering this question. So we're only interested in receiving 1, 1 throughout, and then receiving the control throughout, and then compare their potential outcomes change. But you can also like look at the different treatment combinations. Yeah, like 0, 1, right? Right now we don't we don't estimate that.
More possible, I guess, average treatment effect definition. Right?
Yes. Yeah.
And everybody is going to switch over from that RCT control to.
Not necessary. Yeah.
Not necessary.
Yeah.
It's a possibility.
Yeah. Yeah. But you are making a very good point. So we are actually have some like observations for the RCT control, right? So if they do not switch, but right now we completely ignore such information. Yeah, but leveraging such information actually is useful. Yeah, but this is a very good point. Thanks. Yeah.
You said that MMSE score forms in grooming. MMSE score is a proxy for you that you using the previous slide.
Yeah, so we treated this MMSE as a latent disease severity because they are highly related to the study participation and also the outcome of interest.
Observed MMSE, right? So
in the actual trial, we observed that. But right now, I'm only assessing the methods, so we treat this as a latent. So when the data analysis stage, we do not touch the observed MMSE.
Testing or like evaluating your method, then you will use that.
Yes, yes, yes. Because by this design, we know the ground truth, right? We compare different methods. You will realize that there are so many methods available, so it was very hard to compare the methods on the real data. So the most, at most we can use a plasma simulation to see like different methods, how they work in the real data setting. Yeah, thanks. Other questions? All right. All right, so here we have the existing literature. I would acknowledge that our method is built on a large literature on this problem. Okay, so estimating long-term treatment effect has been of interest for a long time. And I'm gonna outline some main ideas for different existing approaches and see how we develop our methods based on the advantages of different methods. Okay. So the most common and intuitive way actually is extrapolation. So we can feed a parametric model, for example, linear model, to the RCT control outcome, right, data, and then extend the curve into the future. So this is the extrapolation approach. And this is the most common approach in pharma companies nowadays. But clearly, this requires the structural model to be correct, right? So if the structural model is not captured well in the pre-extension period, you can see we have biased result. Okay, so this is the bias of the target. And another one is predictive model. So you can train a predictive model based on the external control. So you can predict what the future outcome looks like based on your covariate, right? And then apply this to the RCT control. Okay. Then clearly it requires the predictive model to be correct. And also it requires all unmeasured confounder variables are not exist, right? And one way to relax the model assumption is we can do non-parametric predictive model. We only have 10 minutes, wrong so fast. Matching is a way that is the non-parametric predictive model, but still we require all unmeasured confounders are captured. And if this is not the case, we have biased result. Okay, so predictive model does not work or require very strong assumption. So the question is how we're gonna leverage tools to handle unmeasured confounder. So here comes the sincere control weighting. So instead of matching based only on the baseline covariates, we can match the individuals from the RCT and external control on not just the baseline covariates, but also pre-extension outcomes, right? Because we have pre-extension outcome observable from both RCT control and external control. So this is the best matching we can do. So this is also sounds very intuitive, right? And if you examine the math a little bit more, it can handle certain degrees of unmeasured confounder, but it requires the unmeasured confounder to enter the model in a linear way. So the underlying model actually is a linear factor model. And if the linear factor model does not work, then the result will be biased, okay? So this is the biased result from synthetic control. So it can not handle study heterogeneity as well. You can see the form of the study heterogeneity must be a constant, okay? And it cannot handle informative missingness, okay? Now, the next class of methods that handles the study heterogeneity in an explicit way. So this is called DID, difference in differences, okay? So this is the classical parallel trend assumption here. So this is, in a plain language means that, so the RCT control trajectory and the external control trajectory evolves over the time the same way. The only possibility is that we have like this constant shift. And the gap here is represented by this Bx plus Bu, okay? So it allows for unmeasured confounder, but it requires a certain form of the gap, right? And this may not be ideal. Okay. And also, if we look at the model, it only allows unmeasured confounder to affect the mean trajectory in a constant manner. Okay, so the unmeasured confounder effect must stay the same way as time changes. Okay, so it cannot handle the case like this. So if we have a linear factor model, then this assumption fail. Okay, so it cannot handle the common linear factor models. In a lot of cases, the impact of latent disease severity actually changes over time. So in those cases, classical DID fails. Okay, so this is a limitation, although it can address the study heterogeneity in certain degree. Okay, and how we're gonna optionalize our assumption on handling unmeasured confounder? We taming the unmeasured confounder through observable proxies. So that's the proximal causal inference. So you can think, although we do not measure unmeasured confounder U, but it leaves traceable traces in the observed data. Okay, so in the sense that we don't have any information about you, but in observed data, you have some information. Okay, so we do this through this the proxies. Okay, so the proxies are important variables that are informative about unmeasured confounder U that satisfy certain conditional independence properties. And a useful metaphor is like this. So proxies are like shadows cast by the unmeasured confounder U. And observing the shadows, we can correct or at least like infer what is the impact of U on the causal analysis. Okay, so that's the metaphor. And in our context, we require two types of observable proxies. So the outcome proxy W, so these are variables related to the outcome, but not trial participation, given U and X. And the examples are like the baseline functional scores or comorbidities not used in trial eligibility. Okay, so there are some guidance on how we're gonna choose the outcome proxies. And the source proxies are variables related to study participation, but not the outcome, given U and X. And the examples including the proximities to the study participation. Okay, and the mechanism here is once we have this outcome and the source proxies, we can bridge between the observed data and the outcome of interest, which is counterfactual. Okay, so how it works in our setting. So for example, here we have the RCT data. For the treated and the control, we can observe the short outcome, but for the long-term outcome, we only observe it from the treated part, but not control part, right? So we're interested in filling these question marks here. And the goal is to predict what YT would be in the RCT control, accounting for the unmeasured confounder U. Okay. And how we're gonna handle unmeasured confounder U, so we define this outcome confounding bridge function. Okay, so this is defined based on observable data, okay, W and X. Okay, and then we define this from the control group, the external control group, which can be learned from using the proxy variable Z. Okay, so once we learn these outcome bridge functions, we only have this. Yeah, then we can show that this is actually connected to the counterfactual of interest. Okay, we can then, under steady homogeneity, we can transport this HWX from the external control to the RCT control and impute all these missing counterfactuals. Okay, but we require these steady homogeneity conditions, and the transportation actually is very stringent if there is steady heterogeneity. So that's why if we directly apply proximal inference, we still get biased result here. Okay, so now this shows the methods landscape. So some method can handle unmeasured confounder like the scenario control matrix completion or proximal inference, and some can handle steady heterogeneity, for example, DID. And there are also existing approaches for handling informative messiness, but you can see when the three challenges intersect, there's a question mark, right? So we don't have a unified framework that can handle all three challenges. And I don't have 5 minutes to talk about the methods, but the key is we introduce this proximal inference framework with a flexible DID, because in the DID framework, we see some restrictions, right? We want to relax that and then develop like causal proximal inference around that. Okay, so we have this capital assumption, small assumption, and also within these assumptions, we develop a doubly robust estimator so that we can leverage the machine learning approaches to predict the nuisance parameters. Okay, so if I do not finish, just remember this slide. And the first assumption we relaxed is the DID assumption. We call this capital, which stands for generalized parallel trends assumption. So we assume that the gap still is a constant BXU, but that's it. Okay, we do not further assume or restrict the form of BXU like to be additive, right? And this is our capital assumption. And translated to the model, it's like this. So you can see we still want the gap to be constant over time, so then we can learn this gap from the extension period and correct this, right? Correct this in the long term so we can recover the RCT control trend from the external control trend. Okay, so that's the key idea. But here you can see we allow U effect to be time varying in this component. So in a lot of medical domain, this is more relaxed than the typical the parallel trend assumption. Okay, and then we assume this small assumption. So this is lies between the missing at random and the missing complete at random assumption. So the missing mechanism is like this. So we allow the RT. So the missing data indicator to depend on U. Okay, so why we call this small? This stands for the missing at random given latent variable. Okay, so it's a powerful middle ground between the MAR and the MNAR. Okay, so that's the mall assumption. And then putting all these three pieces together, we have this proximal DID framework. So we have three challenges, unmeasured confounders handled by the proxies, steady heterogeneity handled by the capital, and informative messiness handled by the mall assumption. And put all this together, we have this causal diagram that shows the assumptions here. Okay, and then we want to define the identification following the causal roadmap we have, right? And it would be very helpful to display the model we assume. So this is the capital model. And then the identification can be done through this, right? So we are interested in this part. So we actually identify three paths toward the identification. One is use outcome bridge function like I illustrated before for the traditional proximal inference. And another one is use kind of like weighting idea. And then the third path is just combine the two, okay? So maybe for the sake of time, I'm just illustrating one identification result. So the outcome bridge function is using the imputation idea as before. You can see the goal still is to predict YT will be in the RCT control accounting for U, but now the steady heterogeneity, I only have 1 minute. So the key idea is that if we directly use the original outcome bridge functions, we get biased results, right? The key is we need to adjust the steady heterogeneity. And where we can find this steady heterogeneity, it's in the pre-extension period, right? So we can define the outcome bridge functions also for the pre-extension period. And what we find is that if you take the difference between the pre-outcome bridge functions for the RCT control and external control, this one actually connects to the study heterogeneity. Okay, and therefore we need this bias correction. Okay, once we do this bias correction, we can uncover review the unbiased RCT control. Okay, so that's the idea. Okay, so what we define this is outcome bridge identification for the parameter of interest. We can also use a weighting idea. Okay, we can similarly derive this identification, and then we can combine both. Okay, and then the combination is very important because one relies on outcome bridge, another one requires a weighting methods, and if we combine them and develop statistical estimators based on this, we can get W robust estimator. Okay, in the sense that we can don't require either model to be correct. If either model is correct, then we can get consistent result. Okay, so that's the idea for the double robust estimator. And then the key here, another piece of interest is how we're gonna learn the bridge functions. So we also borrow some existing literature on this. And the simulation confirms that our doubly robust estimator indeed is doubly robust and applied to the ultimate disease. You can see our proposed method successfully estimated the true long-term outcome, and the existing approaches all have biases. Okay, there are some ongoing work, for example, how we're gonna design the external control, whether you want to deal with the missing data in the analysis stage, or you're gonna exclude all the missing data in the design, right? So maybe like design is very important, but how we're gonna design the external control, so that's some open problem. And how we're gonna select and utilize proximal variables, how we're gonna trade off the simple methods and the machine learning approaches. So these are all very important questions of interest. Gonna stop here. Any questions?
Silly question. So it seems like we have the combined data and we are using one tag and we have these variable age as a part of the tag. But for the RCT data, why should there be unmeasured confounder?
Oh, we're not talking about the confounder for the treatment assignment. We're talking about unmeasured confounder for study participation in outcome. So that's related. You can just think about RCT control and the external control. And the study participation now is the treatment variable.
So that you are showing, so we are not assuming that we have the same tag for both the RCT and the real world data, right? So the tag is only for the real world data?
The tag is only for RCT control and the external control. We ignore the treatment at all, the treatment group.
The second stage, right? So only for the second stage when the control people already went the treatment people in your RCT, like that part.
Yes.
Yeah, I suppose you would have gone into more detail if you had more time. But when you say double robust, what are the two nuisances that would be robust here, right? Like it is robust against, because it seems to me there could be many different nuisances. One is the treatment assignment in the observational population. The other is the S-related propensity score, and then there is some kind of outcome regression function involved somewhere.
Yeah.
There are like multiple nuisances at play.
Yeah.
So what is it doubly robust against? It could be like even multiply robust, right?
Yeah. That's a very good question. So far, I ignored like all the treatment group. So I only focus on the control group. And therefore, there are two nuisances. One is the study participation, and the other one is the outcome function. So, so in the term here, it's the outcome bridge function and the source bridge function. But the source bridge function is just a propensity score function. But this like source bridge function can handle unmeasured confounder. Yeah.
The other question I had was like your S indicator. That's the study participation indicator, right?
Yes.
But like you said, your clinical trial could be small in size, but your observational data like EHR could be much bigger, right?
Yeah. Yeah.
Doesn't that cause like a natural positivity type violation issue for the indicator?
Yeah, this is another problem in our context. So that's another one. So the second line of interest I showed, so that means we have a small RCT, but like large long-term, large like RWD, we want to combine the two data sources. Here we may imagine that RCT is still large, but there's a crossover, and therefore the long-term outcome is not available. And we want to leverage like some information in external control. So size may not be a problem here.
So the two populations are the proportional sizes here.
Yeah.
Comparable.
Yes. Yeah. Yeah, there are a lot of like different data combination contexts of interest. Yeah, sometimes I missed them. Yeah, thanks.
Okay. My question is, I'm also working on some cloud data, but my RCT treatment group size is so small, but I have high dimensional mechanisms like imaging. So there are some external controls. So I'm wondering, if I'm thinking about borrowing information from the external control to my RCT treatment, would that be a idea that makes sense? Because I would imagine like along the time trajectory, although their patients are treatment, maybe there are some like development along the time are still like following the normal trajectory. Part of them are still healthy. If this idea makes sense.
Yeah, it really depends on the study. So if you assume that the external data are relevant, so in the sense that they are free for use, and also there are like domain knowledge that ensures that you can borrow this like natural study, the natural history studies into the treated arm, then you can do that. I think right now the FDA has such guidance, but they also require the domain knowledge to justify this because you don't have any data to check right, whether the external controls are comparable with your RCT treated, right? So you require this like domain knowledge because there's no data at all like to check this assumption. Yeah. Yeah, I think in a lot of like FDA approved cases, they require very comprehensive literature review to justify the use of external data source in this context. Thanks.